Well, thanks again, Dr. Wolf, for taking some time with Julia and I to talk about your research and all the great stuff that you're doing in the field of urobiome research. So I'm just going to kick it off with a couple of questions here. So in your research on the urobiome, how can studies on microbial interactions help uncover the complex relationships between urinary tract microbes and clinical outcomes? Like, how is the microbial interactions actually going to be directly related to clinical outcomes?
Now we know that the bladder's not sterile, and we have a pretty good idea of what species are present. And we're developing sort of network analyses of the communities. So we have some understanding of the different types of communities of bacteria that are found in people's bladders. Now we want to understand how they interact with each other. I mean, ultimately, I'd love to be able to build, an in vitro, right, at the bench community, and then start to probe it. What happens if you introduce a pathogen? What happens if you introduce some, I don't know, some sort of immune response or something like that? But that's really way down the road at this point. And clearly, you could understand that if we can at the bench mimic the bladder in the bladder ecosystem, right, and understand how it can be protective, how it can go awry, right? It can be just in imbalance and then cause symptoms. But to get there, we need to start at the beginning, right? So now we know what organisms are typically present in people's bladders. There's not one type of community, there's lots of different ones, but we do see patterns. And what I like to do is ask the question, well, of the organisms that we see in the bladder, who's good, who's bad? They interact with each other and how they interact with the host. And this is something I've been saying since a graduate student in my lab who left me several years ago. He was the one who first started saying this at his in-house presentations, right? Because he was beginning to answer some of those questions. So the how do they interact with each other is really the question that you just asked.
Right.
We do know that here's an example, right? So a clinician sends a urine sample to the clin lab. The clin lab comes back and says, we found, these organisms, and we have antibiotic susceptibility profiles for each of them, right? Now, mind you, I think that the standard urine culture test that clin labs all around the world use is deeply flawed, okay? But the other thing that they do, because it only gives a partial picture, they're focused in very narrowly on what they think are the important bugs. And I would argue that they're all important to some degree or another. We really need to spend more time thinking about the ecosystem, not who's the top predator here, right? Who's the pathogen? But the other thing that clin labs do is they isolate the bacteria that they think are the pathogens, and then they do antibiotic susceptibility separately, right? You put those two bugs together and ask what's the antibiotic susceptibility of both when they're together. In many cases, it's different.
Is that because the interactions between the two are then
bacteria interact with each other, right?
Right.
So here's a extremely trivial case, but you can get the idea, right?
Right.
So if you take an E. coli that has a plasmid that encodes ampicillin resistance, so it makes the enzyme beta-lactamase.
Yep.
Right? And you put it together in the same culture with an E. coli that does not have the gene for the beta-lactamase, right? It's not ampicillin resistant. They're both insensitive. That both of those bacteria will survive if you throw in ampicillin, if you challenge them with ampicillin, because those of us who know the molecular biology know that beta-lactamase is secreted into the environment. And so the resistant guy is protecting the sensitive guy. Now that's a trivial explanation, a trivial mechanism, right? It wasn't so trivial when they didn't understand that beta lactamases were secreted, but now we've known this for decades, right? But those sorts of interactions that are a bit more complex, they happen. Okay. And so wouldn't it be better to know the antibiotic susceptibility profile of the whole urine sample, of the whole ecosystem? Okay. And wouldn't it be better if you had if you understood how 2 or 3 or 4 organisms interacted with each other, such that the antibiotic susceptibility, you understood why you got the antibiotic susceptibility? Well, your system, your instrument allows, at least the beginning, where we can put 2 bugs together. They're separated by a membrane, so they can't touch each other. You don't have a physical interaction, which, by the way, can happen.
Right.
But they're sharing the same environment. So if one is sending out a signal to the other, a small molecule or whatever, saying, be more resistant, then you can determine, at least that these 2 bacteria are talking to each other and it's affecting their sensitivity to an antibiotic. Then you can, once you have the phenomenon recognized, then you can go in and try to understand the underlying mechanism, and you may be able to use that knowledge to improve your outcomes, right?
Right.
So that's one way that you can do. You could use your system, the interactions. But the other is how do 2 bacteria interact? Forget antibiotics, right? One bacterium might inhibit the growth or even kill another bacterium. And we've published on at least one interaction where one of these urinary microbes is pretty detrimental to most of the species that we tested, right? And it's because it's excreting a small molecule that is just lethal to just about everything other than closely related organisms. Now, we don't understand perfectly how that small molecule works, although we have some ideas, and we certainly don't know how it manages not to kill itself. We don't know what the immunity is, in fact, right?
So how did you guys make that discovery?
That was that was the same graduate student. When he was a master's student, he was one of those that was relatively early on involved in using our enhanced culture method, which we call expanded quantitative urine culture, which shows us what the standard clinical lab test is missing, right?
Mm-hmm.
It was designed after we did DNA sequencing on culture negative, and a culture negatives in quotes because they're not culture negative, right? After we saw DNA evidence of bacteria in what the Clin Lab had labeled as no growth, which a lot of people would interpret as sterile. We looked at the list and said, well, the list of bacteria that we can detect by sequencing are not unculturable. They just don't grow under the growth conditions that the Clin Lab had grown them, right? And so I went to Paul Schreckenberger, who was the director of our Clin Lab and one of our team members. I'm very lucky that we had a director of a clinical micro lab that was very interested in sort of pushing the science along. And I hounded him, in fact, come up with a protocol to grow these bugs, right? So he eventually did. He named it expanded quantitative urine culture, and we co-mentored a master's student by the name of Evan Hilt, who then went on to be a PhD student in my lab, and she's now an assistant professor at the University of Minnesota. So kudos to her. Well, the next guy in, Travis, learned from Evan how to do EQUAC, and so he was very much involved in sort of receiving the samples in the lab. And basically, we were running an uncertified clinical microbiology lab. We couldn't tell the clinicians what we were finding, but we were finding plenty.
Yep.
And he was putting all the data together, and he noticed that, when he looked at a matrix of which bacteria were co-cultured, right? And he made a heat map out of it. He went, wow, whenever I see Lactobacillus crispatus, I almost never co-culture E. coli. I wonder if whether that's real or not, right? So very simple. Grew up some E. coli, spread it on the lawn, let the lawn dry, and then took 10 microliters of a Lactobacillus crispatus
Yep.
culture and spotted it on the lawn, threw it, let it dry, threw it in the incubator, came back next day, and the lawn grew, except where the Lactobacillus crispatus was, the E. coli was dead. Okay. And so he double checked to make sure that it was in fact bactericidal, not just inhibitory. Ultimately, he grew up Lactobacillus crispatus cultures, 48 to 72 hours, spun down the cultures, threw away the cells, filter sterilized this now conditioned medium, this cell-free supernatant, and mixed it together with E. coli and watched E. coli die. So we have, transmission electron micrographs of these just literally eaten up E. coli's. I mean, their membranes are shredded. And so he did a whole bunch of work. I had a couple, he went on to be a PhD student in my group, and he took on a different project. So I had two other master students who came in and proved that what this was something other than what everybody else thought lactobacillus produces. It wasn't a bacteriocin, it wasn't a protein, it wasn't lactic acid and hydrogen peroxide, as everybody thinks that these are the main reasons that lactobacillus is such a good inhibitor of pathogens, right? But it was something else. And finally, we reached the point where I needed a PhD student to take on the project, and I needed a collaborator. So a natural products guy. So I teamed up with Tim Bugney up in Madison, and my student Omar hooked up with his postdoc, whose first name I forget now. And together they went back and forth where we were giving them cell-free supernatant, and they were fractionating it and subjecting it to NMR and basically doing the metabolomics. And then Omar was getting these fractions back, and we figured out which fractions had the activity. Ultimately, we found out it was a small molecule of phenolactic acid, or PLA. So it's lactic acid attached to a phenyl ring, right? And so we think what happens is the phenyl ring intercalates into the membrane and just destroys it, right? But we haven't done the biochemistry, the membrane chemistry to determine that. But we found that PLA, eventually we stopped working with the supernatant, and you can buy PLA.
Yeah.
So we bought it, and we started doing all sorts of things with it. And we found that it killed just about everything except for lactobacillus. And it's not so great on enterococcus, which is related to lactobacillus. It even kills yeast. It kills candida. I mean, it's amazing. And it killed all of these multiple drug resistant bacteria, multiple drug resistant Pseudomonas, for example, that we got from a colleague of ours here. And the thing is, it's stable. So I personally think that some derivative of PLA might be a really good antimicrobial for places where there's no refrigeration. We literally can boil the damn thing and it'll still work. The only problem is it only works at low pH. If you're below 4.5, it stops working. But I think you can probably modify it, move on. And honestly, we would have patented this thing if it wasn't for the fact that somebody already knew about it. But I think it's so, I have no qualms about putting that out there. Someone should turn this into an antimicrobial that's useful. You can boil it, you can leave it on the bench. And this all comes from the kinds of interactions that you're talking about. I mean, he could have made this a whole lot simpler if we had your device, right? Because he could have just put the crispatus in one coli in the other. And the next day there'd be no E. coli there. There'd be no OD. It'd just be debris.
Well, that's a perfect segue into the next question I was going to ask you, which is how we first got introduced. And on the line here, we got Julius Wavola, Dr. Julius Wavola, who is a Cerullo's head of product. And we were at a conference and doing a little presentation and releasing the new technology here. So, Dr. Wolf, if you don't mind, give us a little bit of background on how you actually first became aware of Cirillo and how that interaction went.
Well, it helps to have a graduate student, an MD, PhD student who's actually walks up to a booth and,
Yep.
and asks, well, who are you? What do you have? And he was in fact involved here with a project for another industry partner that indeed involves this sort of antibiotic resistance interactions. And we've been promising them data. And the first 3 parts of it was no problem because of wall and isolation. And then we started putting it together. And my poor research tech, Wilson, was just struggling with how in the world was he going to be able to tell if he put the two together, and he got a change in OD, right? When he exposed them to antibiotic, well, who was contributing to the OD, right? Was it all one bug? I mean, there's a problem, you've got 2 bugs, they grow at different rates, right? So putting them together was just not going to work. It was going to be an incredible amount of manual labor. So since it was Brian who designed the study that Wilson was working on, he feels sort of responsible for Wilson's pain and
His
pain
mental stability at
yeah,
that.
exactly. And so he was aware, he was sensitive to that. He came up to your display and started asking questions. And he ran into me and he said, there's going to be a presentation. You should listen in. And I said, sure. So, mind you, this is a meeting where I'm one of the organizers. So the fact that I was sitting in the back row in the balcony tells you that I guess I was trying to stay out of the public eye for a few minutes. So me and a few of my team were sitting back there. And when the talk was over, what came out of my mouth, subconsciously, unconsciously was, well, that solves all of my problems. And Julia was sitting right in front of me and she turned around and said, I'm glad to hear you say that because I'm part of the company. Would you like to talk to the president? I go, sure. And then what, we spent, I don't know, half an hour, 45 minutes talking over your display until someone dragged me away. So we ordered it up and we've done a little bit of work and it seems to work.
Wow, that is really exciting.
It really does allow us to put 2 bugs in adjacent wells with a membrane in between. And we know that we can measure their growth separately. Once we do that, we can extract the culture out of each independently, and we can do other analyses. One could do, it's sort of cell, the cellular analyses, right? Because the medium's all the same, that they're sharing the environment. But, one could do metabolomics on the culture to see what's now out there, right? So yeah, this will allow us to do relatively high throughput, especially once lots of these duets become available. And a lot less time than what we were trying to do, which was really frustrating us. So yeah, so having a graduate student who is paying attention to his surroundings and a little bit of serendipity, because if Julia hadn't been sitting in front of me, maybe, well, I wouldn't have been introduced, and maybe I would have gone down and talked to you guys, but maybe not. Maybe I would have gotten, dragged away and I never would have managed to get back there.
Well, so, I think that's we'll wrap here, but I think that's a valuable lesson to especially all the PIs who may watch this or listen to this, is not only how important microbial interactions are, but every once in a while you should probably listen to your grad students. They may not have a good idea.
Oh yeah, well, I would say that one of my favorite quotes comes from an actor, a Broadway actor by the name of Harvey Fierstein. Being interviewed on Fresh Air, and I was driving home from work. I literally pulled over to the side of the road to text it to myself so I wouldn't mess it up. But what he said, and I run my lab this way, and I to a large degree, run my life this way. What he said was, life is only as inspiring as the number of times you say yes. Now, in context, what he meant was, sure, I'll take a coffee. Sure, I'll have breakfast or lunch with you. Yeah, let's go have a drink. You can give your elevator speech, and then I'll decide whether or not it's going to be worth my time and effort. And if you don't open the door and walk inside, you don't know whether or not there's a pile of manure or a pile of gold. So spend a few minutes to check out, what other people have to offer. And then you can decide whether or not you really want to sort of team up with them, which is what we did, right?
That's right. That's right.
Right?
So
We invested a little bit of time. It might have turned out, usually it turns out to be nothing, but sometimes there's gold on the other side of that door.
that's exactly right. And
It was a very well chosen seat, and based on my memory, your collaborators were very excited to go out to lunch with you. So I'm not sure they would have let you come
well,
Yeah.
again, thank you, Dr. Wolf, so much for your time. We really appreciate it and look
Sure.
forward to continuing our collaborations and partnerships. And so with that, we'll log off the podcast, and thank you again for your time.